Hello everyone, and welcome to another edition of In Conversation with Nutraceutical Business Review. I'm Dr. Kev Robinson, the editor here, and tonight I have the pleasure of speaking with Eloïse Breton, Director of Product Management at DNA Genotek. Company's made a splash in the area of microbiome sampling and stabilization, and tonight we're going to be talking about driving evidence-based nutraceutical innovation. Let's see what Eloïse has to say. Good evening, Eloïse. Thank you for joining me here today. And I think we're here today to talk about driving evidence-based innovation in the nutraceutical space. And I believe that some of the thinking behind this is your thoughts on the efficacy of some currently available nutraceuticals.
That's correct. Yes. Pleasure to be here.
Could you maybe sort of start us off with what your background thinking is on that and why perhaps the industry needs to do a little better?
Sure. Yeah. So just to put it into context, I've been more on the microbiome side now for many, many years. So I'm coming into this at seeing the microbiome space converging with the nutraceutical space and seeing how that new field is helping understand the potential of nutraceuticals. So that's kind of where my brain is coming from. And what I love about both the microbiome space and the nutraceutical space is that we're going back to that very old concept that humans have known for millennia, which is food is medicine. It's what fuels us. It's what makes us feel good. And, for centuries, we knew which foods could make you feel better if you're experiencing, this type of condition, or you shouldn't eat that if you're dealing with this, or you should eat more of that. And I feel like we kind of lost a lot of that, in more recent decades and maybe even more recent centuries. And now we're going back into that. And nutraceuticals have been around for quite a while. But now we're digging into more of like, what is that mechanism that, that compound that we know, why does it make us feel good? Is that the same effect for everyone, for example? Really understanding the science behind it so that we can better use them. Because I think we've all had stories where, you eat a certain food and you feel fantastic, and then you tell your friend to eat the same food and they try to eat it or consume it, and then they feel horrible. Why is that, right? So it's because each and every single one of us is very unique, and even more so when you consider the microbiome. So I think being able to dig at that science a lot more and get a lot more understanding is just going to prove very beneficial for a lot of us.
For somebody who isn't as familiar with the microbiome, was how the gut microbiome affects health? How does it play that role?
Yeah, so that's a great question, and one that I love to tell even family and friends about, because I think it's really interesting. So I'm going to tell a little anecdote. You can totally cut it out. When I was in school, I remember that in my first immunology class, my professor drew a gut on the blackboard, and he spent the entire semester constantly drawing how the cells of your immune system, which are lying in your gut, are interacting. And so we're learning about, T cells and whatnot. And back then it was just so granular. I wasn't thinking about it big picture. And then, years later, I'm in the microbiome space. And when I did my PhD is when I kind of dug back into the microbiome, and that this concept of, your immune system is in your gut and it regulates everything that happens in your body, and everything that we absorb, all the food that we take in, it gets into our system thanks to our gut. But as humans, we're actually pretty limited in the number of mechanisms and pathways that we can actually carry out to convert the food into the compounds and break it down into the micronutrients that our body is actually able to take up. So that's where the microbiome comes into play. It helps modulate it, it helps bring those nutrients into our system. And not only from the food level, but those bugs that are living inside us, they're also producing compounds because they are consuming the foods as well. And those compounds are being secreted into our body and they're moving around. And through the gut, they then enter the blood. And so they get to circulate everywhere. And so what's been really interesting to learn about over the past decade or so is, that gut-brain axis, for example. We know that what happens in your gut, those compounds, those chemicals that are moving around, they move up to your brain and they can actually modulate how you feel, how if a specific disease state is going to progress in a certain way or not. I've seen it also in the skin microbiome space. People don't talk about, the skin gut axis as much, but it's clearly a thing because there's so many conditions that are like your skin is really just your outer view of your immune system. It's kind of a reaction of what's happening in your gut. And again, that's being modulated by your microbiome. Your microbiome is connected to absolutely every system in the body. Hormones, like I could list them all. So that's why it's really interesting to be able to look at all the health conditions and disease states with a new lens now of the microbiome and saying, maybe the reason why we never fully understood it is because we're kind of leaving out a component. So looking at the microbiome and bringing it all together, because we're essentially just one big system with an engine churning inside us, which is our gut. So being able to bring it all together is really exciting. What else would I add to that?
Well, while you're thinking, as scientists, as researchers, I mean, I'm an ex-molecular biologist, how has it taken us so long to make these connections?
That's why it goes back to what I was saying, right? Like hundreds of years ago, people knew, and we just lost touch with that. And I don't know if it's just, the rise of like industrialization and just the change in how much we're constantly being bombarded by so many other things. But yeah, we've lost touch with our own bodies, and I remember after I found out that I had a gut condition, I tried to change my diet completely, and at some point I was doing a really great job and I felt fantastic. And I went, I didn't know this was how you were supposed to feel when you eat really, really good food, which was just in like testimony to like how I had pretty much always eaten kind of badly. And that was the norm. And so I didn't know that there was something that could be better. And I think we're all kind of guilty of that in modern society. So yeah.
Comparative geographical studies would be incredible.
Yes. And one of the studies that kind of got me really curious about the microbiome, and again, this was when I was doing my PhD, was there's populations in Canada of Somali immigrants who, when they arrive in Canada, start consuming a North American diet. And a lot of them develop tons of like other diseases and conditions that they would have never seen in their home country of Somalia. And some of the mothers kind of tuned into that and started feeding, especially their kids, more traditional Somali diets. And then you see the symptoms coming down. And so that's super interesting. And yeah, I think there's a few people out there who are going into kind of remote communities and trying to get their microbiomes to see what have we lost potentially over the years from, hundreds of years of our current modern diet. Those studies are always very fascinating when they come out.
I look forward to those studies being written up.
Yeah. There's even a great one of it was where they took immigrants who were coming from, I believe they were moving from Asia to the United States, and then they tracked their microbiomes, from just leaving, their home country, and how many weeks it takes for them being in the United States for their microbiome to essentially kind of just be very modified. And it's quite shocking. So it happens pretty quickly. So they're out there already. We can send you some references.
And obviously part of that is where you're helping to drive new innovations here is with your microbiome sampling. Mm-hmm. So tell me, how is it done? And how how is that understanding that individual's microbiome going to play a significant role when it comes to designing more targeted nutraceuticals?
Yeah, so gut microbiome analysis or testing or however we want to call it essentially means that we're using feces as a proxy for the gut. Obviously, we don't want to submit anybody to invasive sampling of their intestines. So we use feces as a proxy to kind of understand what's happening in there. So in the case of our technologies, a donor would produce their sample and then take a small portion of feces and place it into a device such as ours. There's or a tube of some sort. And hopefully there's some level of stabilization there. That's what our products do, because a fecal sample has a huge microbial community that's very active. But since we're trying to understand its general state, we need to stop it and kind of take a picture of what that community is at that time. So that's where stabilizing the sample is very important. And then that sample will go to a lab to be analyzed. And the analysis essentially consists of looking at the DNA of all the different microbes that are present in the sample. We use DNA also as a proxy to kind of say, okay, these were the bacteria that were present in that sample. And that gives us a general kind of profile of this complex community that lives in the gut. And the idea from there is that if we take also all the other data around it. So for example, if I produce a sample and I tell you everything about, my diet, my health status, and all those types of things, and you have all that information in a database, and then you get another sample, another microbiome sample from someone else, you can kind of compare where does that sample lie in relation to my sample, for example. So we're using these huge databases of profiles to kind of tell us this is potentially positive, this is potentially negative. We're also basing that on the scientific literature. And we're ultimately trying to make interpretations to say your microbiome is in this state, and potentially you could improve it by doing these things, or you might make it worse by doing these things. Where microbiome sampling and analysis comes into play in nutraceutical development is at multiple different stages. So you could do microbiome sampling prior to doing a study on whichever nutraceutical you're studying, potentially maybe to understand, is the person going to respond to the intervention or not? We're finding that the initial state of your microbiome actually dictates quite a bit how you will or will not respond to taking something. So that can be part one, to say, is this the right person to be testing with, our nutraceutical? Then it could be during the intervention to see, is the nutraceutical that is being taken having those improvements, or is it having negative impacts? Is it doing the things that we actually wanted it to do? And what is it doing, right? How is the microbiome responding? How are they taking it in, potentially transforming it, and how is the body absorbing it? And then you could even do it at a later stage, potentially after the fact, to see, does the nutraceutical have a lasting impact? Specifically with things like probiotics, obviously, which are active bacteria, most people want to say, well, does it actually stick around, or is it only active so long as I'm taking the probiotic, or is it able to essentially make its home in my gut and then stick around? And if so, for how long? So lots of different areas where microbiome sampling could come in, and all bring a very different level of information.
Fascinating. And I imagine this is where, if you're taking samples, say, beginning, during, or after a trial or an intervention study, this is where the stabilization really plays a key role, because it's at that moment. If for example, your sample is, like we have to do in the UK, sometimes put it in the post,
Yep.
It could be days later before that is analyzed, and you could have a completely different result.
Exactly. So we really need to stop the sample at that time to make sure that the bacteria don't continue to grow, because they are essentially in their food source. They're very happy, so they could continue to grow and completely change their ratios. And we've seen it. Tons of researchers have demonstrated that the sample changes immediately. And that's why being consistent and ensuring that the sample is always taken the exact same way also becomes very important, because if you take a sample, one day and you take it in such a way and you take it differently another day, the results are going to be completely different. So we really do need to standardize quite a bit. And that's true for sampling and all of the steps that come after the fact, whether it's how the sample is extracted, how it's sequenced, and also how it is bioinformatically analyzed. Like if you're not using the same tools, if you're not being consistent, then it's very hard to make sense of what you're seeing. You're just kind of trying to interpret gibberish almost, if I can put it in a very crude way. So yes, every step of that process is very important.
Okay. And tell me a little bit more, if you would, about the analytical stage, because we must be getting through an awful lot of samples, creating a lot of data, and maybe needing quite different analytical techniques to be able to really understand what the tests and the results are showing.
Yes. And I think one of the reasons, I don't know if it was just serendipitous or if it was causation, but the fact that we're living through an AI kind of revolution right now and that we're seeing so much more machine learning is timely, because we really needed that to be able to take microbiome science to the next level. There's only so much that our human brains can kind of look at and try to analyze without putting these very, very complex samples and data sets into those types of tools and helping us interpret the results. And one of the issues with microbiome science is that there is so much data and there are so many variables at play. So that data is just, it's huge. And to be able to actually get something out of it, we need those tools to be developed. We need them improved. And, I spoke about purely the what we call quote unquote microbiome data, which is the traditional microbial profiles, but we're now seeing the potential for adding metabolomic data, metatranscriptomic data, which is way more informative when you combine all these data sets. But then you're okay, well, how do we interpret them? And we need the tools to be developed at the same speed to help us get there. But it's timely. Things are working out well for the space, so we're moving in the right direction.
It was one of the questions I had as an aside. Do we have more data that we can cope with? Is the technology keeping up with the amount of information that's being put into these data sets to be able to discern the true result? And maybe like that's happening in clinical trial data these days. Is there a potential that in the future, we may find stuff that we weren't even looking for when we further analyze it?
I do expect that's 100% going to be the case. And that's, again, going back to something I said before, why standardization from the get-go is so important, because those samples that we analyzed, 5, 6, 7 years ago, the sequencing data that's sitting in somebody's, hard drive, we want to be able to upload them to these others, combine all these data sets and be able to analyze them. That's where the power truly is. And I don't think we have too much. We probably have too much data maybe for the tools that we have today. But you can't have too much data. The more data, the better, right? And we've seen it, DNA Genotek, my company, we come from the space of human genomics, and we saw the same thing happen where if you had your genome analyzed years ago, they could tell you X, Y, Z. And if you have that same genome reanalyzed, 5, 6, 7 years later, they're telling you completely different things, because so much more data has come into the system and it's coming out, and it's much more informative. So we're going to see the same thing from the microbiome space for sure.
Fantastic. And I guess a few years ago, getting your genome sequence would have cost millions of dollars.
Yep.
And these days, it's probably quite run of the mill.
Yeah, exactly. So that's the other thing, is that the more the technologies become mainstream, the better the costs become. And then so we can do a lot more of them. I mentioned metatranscriptomics, which instead of just looking at the microbial DNA, you're looking at the microbial RNA, which is telling you what they're doing rather than who is there. They're really, it's really telling you they are doing, they're using these mechanisms and they're active in this way. But metatranscriptomics is still quite expensive to do, so it's not as common, and it's so new that, again, we need to develop tools to be able to interpret that data. So it all has to come together. So,
Sure.
But it's coming.
Good. And what about epigenetic factors? I mean, obviously, if somebody, whatever they're taking nutraceutical, if they're, say, stressed or something,
yeah.
We know that the gut is linked to every aspect of the body now.
Yep.
Can you edit that out, or is that just something that kind of goes along with analyzing the data in the end?
That's something that I'm hoping we're going to see a lot more of, is taking into consideration both the host genetics, host epigenetics, and then potentially also what's happening in the microbiome. And that goes back to what I said, that there's so many variables at play, right? And you can see it on a short time frame or a very long time frame. And that's the other thing, because just in the microbiome space, for example, we've been able to show that if you leave your house, take a flight, go to a different country, come back, all of those actions change your microbiome within a short time frame. So what happens also, like long term, if you're seeing all these different changes, or if you're exposing your body to stress, for example, we don't know, because we're not tracking all of them. But there's tons of, companies and startups that are now saying, okay, well, we've got all these technologies where, you can have your watch tracking pretty much everything that you're doing, and your phone is tracking everything that you're doing as well. So how can we use those data sets to also inform potentially genomic data and microbiome data to get to an even better understanding?
Fantastic. And have you seen any ingredients that you might be able to talk about, or products that are showing really good evidence of efficacy?
There's a couple that come to mind. In the probiotic space, it's a little bit more tricky. I think, those ones. For one, the one that pops to mind is Akkermansia. There's always tons of people talking about Akkermansia and its potential, and it was kind of like established as one of those that, that this is the bug. And now I'm just like, okay, well, I love hearing updates and new presentations around that to really see, have we tapped into what we thought it was going to do? I think of simple ones on the more traditional nutraceutical sides, like turmeric or curcumin, right? That has this anti-inflammatory abilities. Like, I would love to dig into that and understand, how is it so effective at being so anti-inflammatory? Is it that like microbiomes love it? And which microbiomes? Is it a particular portion? And how is there something that people need to do before they start taking, tons of turmeric or curcumin to actually reap the benefits of that? So the little traditional ones, the ones that have been around for hundreds of years or even thousands of years, are the ones that I think that's really interesting, because our ancestors clearly knew something, and we should, like, I want to know what it is exactly, because as a scientist, I want to know these things. So yeah, for me, it would be those ones.
I would agree. Yeah, it would be to give them, there's always proof of concept or proof of use or historic use, but to underline that with, we now have all this supporting scientifically validated data to back it up, would be incredible.
Yeah.
And I think that would take a lot of the, and people still use the term snake oil, but it would take a lot of that doubt away.
Yeah. And I'm blanking on his name now, which is absolutely horrible. But there is a fantastic researcher in the microbiome space who always talks about traditional Chinese medicine and about the fact that, ancient Chinese people knew that, this was good for that and this was good for that. And again, I think we've lost that along the years, because we're like, well, it's a grandma's tale, it's an old wives' tale, and nothing was true about it. But if they were using that for hundreds of years successfully, there had to be something behind it. And now, we all want that scientific proof. So, and a lot of people are going back and saying, yep, let's look at those, those things that people used to use hundreds of years ago, and find there had to be a reason why it worked, and we can find it.
Very much so. And in this age of ultra-processed foods, I think that's top of many people's minds right now.
Yeah, absolutely. And that goes back to what I was saying at the beginning about food is medicine, right? We have to remember that. And when you think of an ultra processed food, what is that doing to your body versus what your body actually wants? And simple things have also come out of microbiome research, just like fiber, super, super good for you. Like we need a lot of fiber and we're probably not eating enough of it. So being able to pay attention to that and how we can be better at doing that, it'll be good that all of this is coming back hundreds of years later.
And to be able to provide a decent sample for your testing, fiber is going to be important.
Yeah, absolutely.
Eloïse, we've covered an awful lot. It's been absolutely fabulous. I think just looking ahead, you've covered so much, but do you anticipate any further developments where we might see more of an intersection between nutraceuticals and the microbiome?
Undeniably, yes. We will see a lot more microbiome kind of leaping over into the space. It's just how is it going to be used? That's where I kind of am most intrigued to see. Earlier, I touched on the fact that, you can do your sampling prior, during, and after. And I'm really curious to see how people are going to integrate that more. On the more kind of traditional pharmaceutical side, the the analyzing of the microbiome pre-intervention is becoming a real hot topic because we're seeing a lot of failures in the pharmaceutical space. And, you look at the studies and you go, actually, it's probably because you didn't select your participants well enough based on their microbiome, that you're seeing that failure. It wasn't that your compound is bad. It's just, it's potentially not right for these people. So seeing maybe more of that is something that I would be very curious to see if it already makes that crossover. Because obviously it's a hard kind of pill to swallow for a developer or, a researcher to look at a population and go, oh, we can't use, that population because they're not going to respond as well. And you also don't want to be seen as kind of skewing the samples and biasing them. So it's positive for you. So that's a tricky one. But then it also brings into play, should we be testing ourselves or should the consumers at some point be testing or using these platforms to inform themselves about their own profiles so that they can kind of look at a probiotic or another nutraceutical and decide, oh, I'm not that right profile, so I'm going to pick something different. I think we're going to, like I always say, I hope that when I'm, 80, 90 years old, it's very commonplace that everybody's going to be taking samples of their feces so that they can use them to either inform, their doctor or how they're going to eat. A lot of people tell me that's far too pessimistic. It'll be here far sooner than that. But I guess we'll just have to see how quickly we can bring it in for sure.
I think we'll probably see AI. You take the test, analyze it at home, and then some AI bot will tell you, "Hey, you need some omega-3s or you need some prebiotics," and that's hopefully where we'll be going.
Yeah, that's what I like. Again, I think it's just the timeline question. Is it going to be in the next 10 years or is it going to be in the next 30, 40 years? Only time will tell. But it's a very exciting time to be in the space.
Thank you very much. And Eloïse, the sands of time are sadly ticking out. So I think if we're all happy, we'll call that a wrap.
Yeah, no, that's great.